So, dear students, today we have next lecture about protein synthesis, and today we will integrate knowledge from uh, knowledge about genetic material and knowledge about what happens outside of the nucleus, what happens in the cytoplasm in order to get protein synthesized. So let's begin. And we will begin from this simplified illustration of one chain of the DNA. And as you already know from our previous lecture, each three nucleotides that we call triplet encode one amino acid. And amino acids arranged in a chain will make primary structure of the protein. And now let's project this information to illustration of the cell. So here you see a cell with nucleus, so DNA that contain information about order of amino acids in the protein. So this information encoded inside of the DNA, and this DNA located in the nucleus. So it is safely stored in the nucleus. However, protein should be synthesized inside of the cytoplasm, not inside of the nucleus. And this is schematic illustration of the protein that is synthesized on the ribosome inside of the cytoplasm. In simplified way, we can say that ribosome is kind of production department, while nucleus, once again in very simplified way, is kind of information department. And what we have to do, we have to transfer information from information department to production department. But we are not allowed to take DNA out of the nucleus. In order to make this job, we need a special transport system. And of course, if we will use different portions of DNA with different information inside, we will have different structure of the protein. So this is example when, let's say, one portion of DNA with information about protein 1 was used to construct the protein. And this is example when another portion of genetically encoded information, information about, let's say, protein 2, was used to construct another structure of another protein. So how we transfer information from DNA that is located in the nucleus to ribosome that is located outside of the nucleus, inside of the cytoplasm. So this is very important questions that have to be addressed. And this is achieved with the help of messenger RNA. Sometimes you may find another term, informational RNA, but messenger RNA is more common. So, information is transferred from DNA that is located in the nucleus into messenger RNA that initially also located inside of the nucleus. And then mRNA is allowed to escape from the nucleus into the cytoplasm, and it is transferred inside of the ribosome. And now we have production department equipped with information about the structure of the protein that have to be produced. And as you see, now we have primary structure of the protein produced by the ribosome on the basis of information that was provided by messenger RNA. And when primary structure of the protein that composed of chain of amino acids is completed, this chain of amino acids will be disconnected from ribosome. And then will happen another very important event, because usually primary structure of protein is not an active protein yet. When it is in the form like you see, primary structure, it is not active. In order to acquire specific functional activity, this primary chain of the protein have to be arranged in special three-dimensional conformation. For example, it have to be transformed in such a half-circular structure. And now you see that due to three-dimensional conformation change, inside of this structure there is kind of active center that can be used for chemical reaction with another components. So this is very important step that is called post-translational modification. During this step, protein will acquire special three-dimensional structure that is necessary for its normal functional activity. And now let's summarize these steps. First, have to be performed transfer of information from DNA into messenger RNA inside of the nucleus, and this is called transcription. When information that is encoded in messenger RNA is used by a ribosome to construct primary chain of the protein, this is called translation. And three-dimensional confirmations of acquisition of special three-dimensional conformation by chain of the protein is called post-translational modification. Another important point that have to be considered during protein synthesis is transport of amino acids from the cytoplasm into ribosome. Amino acids are building blocks of the protein, and they distributed inside of the cytoplasm. However, you know that proteins synthesized with the help of ribosomes, and this means that you need special transport system that will bring amino acids from the cytoplasm to the ribosome. And this transport system that is necessary to move building blocks of the protein inside of the ribosome is called transport RNA. Connection of specific amino acids with corresponding transport RNA is called activation of amino acids. So once again, to summarize, activation of amino acids is connection of specific amino acids with corresponding transport RNA that is necessary for transfer of these amino acids to ribosomes. And another important point that I want to highlight is that each amino acid has its own specific type of transport RNA. So one transport RNA that is responsible for transportation of, let's say, serine will not be connected with another amino acid, let's say proline. For serine, there is one highly specific type of transport amino acid. For proline, another one. For tyrosine, yet another one, and so on. So once again, to summarize everything, protein synthesis composed of following steps. First is transcription, next one activation of amino acids, translation, and post-translational modification. And now I want to draw your attention to this simplified illustration of information flow inside of the cell. Yeah?
May I ask you something? How the
Please,
mRNA?
please write down your question and we will discuss your questions at the end of the lecture. It is highly likely that during my lecture, I will provide you with answers. So first, I will give you a lecture and most likely you will find your answers during my further explanations. And
Okay.
at the end, I will be open for questions. So here you see a simplified illustration of what we call information flow inside of the cell. And it's very simple with only few steps. Information flows from DNA into RNA and then through the RNA to a protein. Transcription corresponds to this step and translation corresponds to this step. Now let's look what happened during transcription in more details. Transcription is a process of mRNA synthesis on the matrix of DNA. And this is this step, information flow from DNA to messenger RNA. Let's imagine that we have here a chain of DNA composed of nucleotides that you see here. Thymine, adenine, thymine, guanine, cytosine, thymine, cytosine, cytosine, and adenine. And our task is to transfer information encoded in this piece of the DNA into the form of RNA. And now comes very intriguing question. What should we write here? What nucleotides, what should be specific sequence of nucleotides in RNA if we know sequence of nucleotides in the DNA? And this is very important question. I would say this is one of the key questions of genetic code. A code that is used by the nature in order to perform protein synthesis. In order to, let's say, describe how proteins should be synthesized. So you see that we have kind of a challenge. We know what is the sequence of nucleotides should be in DNA, but what should we put inside of the RNA? Let's imagine that you have a computer and you want to transfer information from your computer to your USB stick. And in order to make this transfer of the files from your computer to USB stick correctly, your computer and USB stick have to use certain information transfer protocol. Otherwise, if information transfer protocol will not be used, your file on the USB stick may be not readable, may be damaged, it may be not possible to open it. So without correct data transfer protocol, copy of your file may be not correct. And this may lead to such situations that you will not be able to use it. That's why here, if we will not follow certain data transfer protocol, and if we will make a mistake during transfer of information from DNA to RNA, this will lead to mistake in the sequence of amino acids inside of the protein. And this in turn will lead to such situations that protein that will be synthesized with mistakes in its structure, such protein will not work. And for some proteins, this may lead to dramatic consequences for health of the patient if some proteins will be produced in not normal, not functional way. So what is data transfer protocol that have to be used when we want to transfer information from DNA to RNA. And now we have to come back to a topic that we discussed during lecture about DNA. During that lecture, I told you that in the structure of DNA, thymine nucleotide corresponds to adenine nucleotide. And this is part of complementary principle, that thymine corresponds to guanine, guanine corresponds to cytosine, and for DNA RNA interconnections, this complementary principle is slightly different because inside of RNA there is no thymine, and instead of thymine there is uracil. I hope you remember this, and I told you that this is something very important. So this complementary principle that tells us that thymine corresponds to adenine, guanine corresponds to cytosine, and adenine corresponds to uracil is data transfer protocol that is used by the nature in order to perform transfer of information from DNA to RNA. And later you will see that same data transfer protocol will be used to transfer information from RNA to protein. So these two critically important events in the life of the cell that are related to synthesis of the protein, transfer of information from DNA to RNA, and then from RNA to protein, guided by very simple data transfer protocol that is presented by complementary principle. So when you know that thymine corresponds to adenine, let's find thymine nucleotides in our DNA. They are here highlighted in red color. And this means that corresponding nucleotides in the RNA should be adenines. Now let's find adenine nucleotide in our DNA, and according to complementary principle telling us that adenine complementary to uracil, now we know that corresponding nucleotides in the RNA should be uracil. What about guanine and cytosine? Let's find guanine. Corresponding nucleotide in the RNA will be cytosine. Cytosine in the DNA will correspond to guanine. You see, now we performed correct transfer of information from DNA to RNA with the help of data transfer protocol represented by complementary principle. And now let's look in more detail how this happens. Here you see simplified illustration of double-stranded DNA, and in order to synthesize RNA on the basis of DNA, these helix have to be splitted. Yeah, so these two chains have to be locally splitted. Something like this. And then, with the help of special, and now let's imagine that inside of this chain, we have following nucleotides like adenine, thymine, guanine, cytosine, adenine, adenine, and so on. They are grouped here for triplets for simplicity of our explanations. And then a special enzyme that is called RNA polymerase will attach to DNA, and then it will move stepwise through the DNA, and it will synthesize RNA on the basis of sequence of nucleotides that is present in DNA. You see, with the help of complementary principle, this guy is simply adding new nucleotides to the growing chain of RNA, And at some point, this enzyme will stop, and now we have new, freshly created chain of messenger RNA that was created on the matrix of DNA with the help of complementary principle. Then this enzyme will stop, and this RNA will detach from DNA, and two strands of the DNA will be closed again in this structure. So what we just done, we created messenger RNA on the basis of information that was written in certain portion of DNA. And this is what we call gene. Fragment of DNA molecule that encodes primary structure of the protein. You see, so we did not perform transcription from all lengths of the DNA. No, we want to synthesize only one protein, and for this we need information that is encoded in certain limited region of the DNA that is responsible for this particular protein that we are interested in. And fragment of DNA molecule that encodes primary structure of the protein, this is what we call gene. Later, we will discuss another concept of gene that tells that gene is unit of genetic information responsible for development of certain property of the body. This also one of possible explanations when we consider development of whole body. But now, when we are inside of the cell and we're talking about synthesis of the protein, we will follow this concept of the gene. So that gene is fragment of DNA molecule that encodes primary structure of the protein. And very important and very surprising trick of the nature is that gene includes not only nucleotides encoding sequence of amino acids of the protein. Fragment of DNA that is responsible for information about synthesis of the protein also encodes nothing. So there are some regions in the gene that we call exon, and these are coding region of the gene. These, I would say, sub regions of the gene that encode information about sequence of amino acids. This is what we call exon. But at the same time, inside of the gene, there are so-called introns, and they are non-coding regions of the gene. This is how it looks like. DNA strand. Here you see a fragment of DNA that corresponds to a gene, and inside of this gene, you see fragments of DNA, exon 1, exon 2, 3, and 4, and these fragments of the gene, they encode information about structure of the protein. But between them, you see introns, and introns are fragments of the DNA inside of the gene that not encode structure of the protein. So they are located there, but they not carry any meaningful information about structure of the protein. And question why nature spent an effort to put these introns inside of the exons? And another question, what should we do with these introns if for construction of the proteins we actually need only exons? So nature spent an effort and gave to us something that we will not use. And the question is why it is like this and what should we do with this? And in a few seconds, you will see answers to these questions. So first of all, when we perform transcription of messenger RNA, we transcribed exons and introns. And our messenger RNA at this particular time point, immediately after transcription, is actually not ready to be used for protein synthesis because this messenger RNA has these introns. That's why we call such messenger RNA that is not ready yet, we call it pre-mRNA. So premature or not yet mature messenger RNA. And then this premature messenger RNA have to be transformed into mature messenger RNA. So we are not allowed to use this premature messenger RNA outside of the nucleus for synthesis of the protein. We have to transform this premature messenger RNA into mature form. And this process of transformation of premature messenger RNA into mature messenger RNA is called processing. And processing composed of 3 steps that we call splicing, capping, and polyadenylation. Let's speak about them one by one. Splicing. What happens during splicing? Here you see a schematic illustration of premature RNA with exons and introns. And one of our obvious tasks is to get rid of introns. And this is what happens inside of the nucleus during processing of premature messenger RNA. Introns will be cut out. And then we will have several exons. Each of exon encode information about structure of the protein, and we need all of them in order to get full information about structure of the protein. That's why we have to splice these exons together in one chain. And it will look approximately like this. In very simplified way, this illustration shows you important concept. Introns cut it out, and then those exons that we have, they will be spliced into one chain, and now we have chain of RNA without introns having only important information that encode sequence of amino acids inside of the protein. But what if we will play a game with these exons and we'll arrange them differently? For example, let's put splicing 3 immediately after, let's put exon 3 immediately after exon 1 and exon 2 before exon 4. Is it possible? Yes. And this is what we call alternative splicing. You see, with the help of this game, with cutting out introns and different arrangements of exons from one limited region of DNA, we can get several different messenger RNA that give us several different proteins. This mechanism of introduction of introns greatly enhance information capacity of DNA, because with the help of this mechanism, from one gene, from one region of DNA, we may create several different proteins. And this is a trick. With the help of this different arrangement of exons, we can create several different proteins. with the help of splicing and alternative splicing. But this is not end of the story with maturation of RNA. Of course, now after the splicing, we have RNA that carry only important, meaningful for protein synthesis information, but it is not yet ready to leave the nucleus. Because if it will leave the nucleus in such way, without protection, it will be destroyed by the enzymes inside of the cytoplasm. It will not reach ribosomes because it will be simply destroyed. In order to prevent this destruction, we have provide additional protection for this messenger RNA before it leaves the nucleus. And this is what happens during capping and polyadenylation. So what is capping? Capping is modification of one end of messenger RNA, and polyadenylation is modification of another end of messenger RNA. And now, after splicing, capping, and polyadenylation, we have messenger RNA with meaningful information that is protected from destruction inside of the cytoplasm by the enzymes. Mature RNA leaves nucleus and is transported through cytoplasm towards the ribosomes. And capping with polyadenylation protects messenger RNA from degradation in the cytoplasm. And now we are ready to start next step that is called translation. Once again, what is a translation? Translation is process of construction of the protein inside of the ribosome with the help of information provided by messenger RNA. And translation composed of several steps: initiation, elongation, and termination. And now let's imagine that we are inside of the cytoplasm looking at ribosome. Here you see ribosome, and we see, oh, it's moving. And we see here messenger RNA. So this messenger RNA arrived here from the nucleus, and it was associated with ribosomes, with certain ribosomes. Okay, what else do we need to construct a protein? We have information, we have kind of production unit that will be used to construct a protein. We need additionally building blocks. Building blocks that are represented by amino acids. Here you see simplified illustration of amino acids that are located inside of the cytoplasm, and in order to transport this amino acid into ribosome, they have to be associated with transport RNA. Transport RNA in very simplified manner can be illustrated with this picture. And important point that on top of this transport RNA, there is a special and highly important region that composed of 3 nucleotides. And this is what we call anticodon. 3 nucleotides in the messenger RNA represent triplet or codon. So one triplet or one codon provide us with information about one amino acid. And here, inside of the transport RNA, there is an anticodon that is used for synthesis of the protein. For simplicity, I will skip this long structure and will use illustration like this. You see, this picture shows to you that this particular amino acid is associated with transport RNA that has anticodon with three following nucleotides: cytosine, adenine, adenine. Another amino acids, they are associated with another types of transport RNA. I told you before that each specific amino acid associated with special corresponding type of RNA. For example, if this amino acid is associated with transport RNA that has anticodon, cytosine, adenine, adenine. Another amino acid associated with another type of transport RNA that has anticodon, adenine, uracil, guanine. And if this transport RNA has anticodon, adenine, uracil, guanine, it will not be associated with this amino acid or this amino acid. So, to summarize, each amino acid is associated with highly specific transport RNA that has anticodon corresponding to this particular amino acid. And now let's look how all this system is working. So you see here triplet of nucleotides, uracil, adenine, cytosine. And again, according to our data transfer protocol or complementary principle, we are allowed to put here only those amino acids that associated with transport RNA that has anticodon complementary to this codon. You see, so we are not allowed to put here anything what we want just by random choice. No. We have to look what information is written inside of messenger RNA. We have to look at this codon, and we are allowed to put here only amino acid that is associated with transport RNA that has complementary anticodon. And if our codon uracil, adenine, cytosine, complementary anticodon will be adenine, uracil, guanine. You see, again, we are using complementary principle, but now for transfer of information from messenger RNA into protein chain. What next? Next triplet of nucleotides or next codon is guanine, uracil, uracil. And once again, we are allowed to put here inside only amino acid that is associated with transport RNA that has anticodon complementary to this codon. And this will be this one. You see? Guanine complementary to cytosine, adenine to uracil, adenine to uracil. And now we have here inside of the ribosome one amino acid and another amino acid. What will happen next? Next, this guy will be transferred here. Okay, and connection between them will be established. Okay, let's do it in simple way like this. And now this transport RNA is not needed more in this region. It will be moved out. What will happen next? then will be a movement, and we have now here a space for new amino acids. And once again, we are allowed to put here only amino acid that is associated with transport RNA that has anticodon complementary to this codon: cytosine, guanine, adenine. This one. What will happen next? I think you can imagine. Now these guys will jump here, and you see that now we have repeated steps that will lead to growth of the chain of the protein. And we know exactly what specific amino acid will be next. Next specific amino acid will be the one associated with transport RNA that has anticodon complementary to this codon: uracil, cytosine, guanine. This one. And same story will occur again and again and again. Until the moment when a ribosome will reach so-called stop codon. Stop codon is a triplet that not encode any amino acid. And then all this beautifully organized but not yet functionally active primary structure of the protein will be released. What will happen next? Next will happen so-called post-translational modification. Inside of the cell, there is a so-called endoplasmatic reticulum. And this primary structure of the protein will be transferred inside of endoplasmatic reticulum. There, inside of endoplasmatic reticulum, this primary linear chain of the protein will acquire special three-dimensional conformation. Something like this. Okay, now we have special three-dimensional conformation of the protein, but this is also not the end of the story. Imagine that you created something by your hand, and then you have to deliver this product to the place where it will be needed. What you have to do? You have to bring this product to post station, and there your product have to be packaged and then sent to the place where it is needed. And such kind of post station responsible for packaging is called complex Golgi. This protein, let's make it smaller. This protein will be transformed from an endoplasmatic reticulum. And by the way, this endoplasmatic reticulum that is associated with ribosomes is called granular endoplasmatic reticulum. Granular because a lot of ribosomes associated with endoplasmatic reticulum looks like dots or like granules. That's why this structure, endoplasmatic reticulum, that is associated with ribosomes and involved in protein synthesis, is called granular endoplasmatic reticulum, in contrast to smooth or agranular that is not associated with ribosomes. So this protein will be transferred from endoplasmatic reticulum inside of the complex Golgi. And there, this protein will be packaged inside of special transport vesicles. And this is very important because let's imagine that we created protein that is responsible for destruction of another proteins, like digestive enzyme. And if this digestive enzyme will become active immediately here inside of complex Golgi or active immediately somewhere in the cytoplasm, it will start to digest cell from inside. That is why for transportation and for protection of the cell from activity of the protein, this protein will be packed into a special transport vesicle. You see? Now we have a protein that is packed in transport vesicle that will be used for further transportation of this protein to a place where it is needed inside of the cell, or maybe it will be used to transport this protein outside of the cell. And at the same time, this transport vesicle will protect cell from activity of this protein in wrong place in wrong time. Because in order to be useful, protein have to perform its activity in correct way, in correct place, and in correct time. And this is the end of our story about protein synthesis for today. I hope that this lecture help you to organize your knowledge about protein synthesis from separate pieces of information into one systematic picture. Thank you for being with me during this lecture, and we will meet on our new lecture